About for your next summer winter break. You can help rehabilitate rescued elephants, track endangered rhinos, build a new house for a homeless family, work at an orphanage, teach at a community school, or scuba dive every day to help build the largest man-made coral reef in the world. You're making a huge impact in culture across Africa, South America, and Cambodia, working with families, communities, and wildlife. And if you're more adventurous, you can track to Machu Picchu, dive with dolphins, live off a tropical island. So you guys are up to the challenge. Grab one of these orange flyers and pass it around now. They have an information sessions on Thursday at 12:02 and 3 p.m. in the SAC. Ignore the date of the flyer. It's wrong, my bad. But it's this coming Thursday. So if you can make it, great. If not, check out the website at the bottom of the flyer. So thanks for your time, guys. Enjoy your class. And thanks, Professor.
Also very, very relevant to you guys. The first VBS meeting will be tomorrow at 7 p.m. in the conference room. So we hope to see you there. We'll have food. We'll have more information about our upcoming year events. Also, more importantly, if you haven't gotten your 221 lab notebooks, Ms. Shant will be selling them right after class, right outside those doors. And you can get them from him.
Woo! Woo!
All right, great. Can everyone hear me in the back? Can you hear me? Okay, great. Let me know if this starts to conk out partway through, but I've got a green light for now. Great, welcome back. And there's another event this afternoon for DME over in the SAC kickoff event. I'll be there. Hope to see many of you there at 4 o'clock this afternoon. All right, so let's get started. I know there's a lot of social events now to discuss rhinos and swords. Great stuff. I need your attention here. All right, so we're back for lecture 2, and I just wanted to give you guys a chance to ask any remaining questions and to remind you, I have to remind you that the syllabus is posted online, so lots of detailed information there for those who are not here and missed the first class meeting. As well, lecture notes for the first class are online. Also, problem set 1 is now available online. You should be able to access it. And it is due next Tuesday at what time? Noon, right? So before this lecture. If anyone is having trouble accessing Canvas, you can't find the files, please talk to your TAs. We've all been able to troubleshoot some problems over the weekend, and we can help you get started. Are there any questions about the class or materials that were on the syllabus now that you've had a chance to peruse those a bit more? Yes.
Do you prefer to type it as a word or could you write it and scan it?
You may either, that's a good question, you may either type or handwrite the answers to the homework. If you handwrite them, you have to scan it back in as a PDF. If you feel like your handwriting is neat enough for your TAs to read, I'm happy for you to do that. Either way works for me. There were a number of questions about recording the class. Some of you noticed that one of our learning assistants was recording the last lecture, and there were many people wanting to know, will that become available to all of you? The answer is yes, that will be available. I will give you a bit more detail about how to find that when we figure that out, maybe in the next day or so, so give him a chance to work out the technical details. But yes, we plan to make all the video lectures available to you, and equally everyone will have access. So it's come to my attention that these are some premium ticket pricing for physiology lectures, so all those will be available on the web. Any other questions, operational questions before we begin? A lot of you are emailing me. It would be really helpful when you email if you put the course title in the subject line. It will immediately get flagged as like very important business. So do that instead of labeling it as a question, and I'll get back to you just as soon as possible. Okay, great. Well, we have a lot to cover, so let's get started. And I think if you spend some time with your physiology textbook over this long weekend, you're probably starting to realize that the first, I would say, 1 to 4 chapters contain quite a lot of background material. And it will be material that's going to be important. We're going to be using it again and again throughout the course. Some of it you may have seen elsewhere before in other classes. For some of you, perhaps that was long, long ago, like in AP biology, in high school, depending on what kind of biology you've taken most recently. And for some of you, maybe you already have that material at your fingertips already. If that's the case, I congratulate you. I assure you that will not be the case for most of the course. But this lecture, we're going to have a good deal of backgrounds to bring everybody up to speed and to look at some of this background information in a new way, a bit distinct from what you've seen in other classes. All right, so outline for today, we're going to talk about some ground rule definitions, why are we here, what are we studying. We're going to get really deep into the levels of organization. We're going to talk about size scales and biological systems, building blocks of biological systems, how to build cells, how do cells build tissues, how do those tissues assemble into organs and organ systems. So that's already quite a bit to cover. Then we're going to get a bit deeper into types of cell-cell contacts, cell extracellular matrix contacts. As well, we're going to cover types of tissues. And then at the end, we're going to get to a case study that hopefully pulls together, a case study that pulls together a lot of this information in many of those levels. And we're going to try, at least in all the lectures that I teach, to have a case study in each one, because I think that's a really fun way and a unique aspect of physiology. So we can talk about what goes wrong as well as how normal processes work. So physiology is a little different from anatomy, right? We will cover a bit of anatomy, but physiology, here's some of the definitions from the dictionary. Science of the normal functions and phenomena of living things. Normal functions of living organisms. We can take the Wikipedia definition, study the mechanical, physical, and biochemical functions. A word that comes up over and over again, functions. We're really here to study, to emphasize the function of organisms, their component organs, the chemical and physical processes that contribute to those, how those systems work. And so a little bit different from anatomy, where the emphasis is really on the structure, is on just the organization. It doesn't necessarily link that to the function. Certainly anatomy is something for the approximately 30% of you that go on to medical school, you're going to get really deeply into. But in this course, you're actually going to get a great introduction to anatomy, I think, to anatomy and histology, which is starting to understand how the component tissues are organized, how to recognize important features of those components. And of course, all that is really tightly linked to human medicine, where you get to think about not only how the normal processes work, but pathophysiology, diagnose and treat malfunctions. So all those are very tightly linked. And a major theme of this course is really going to be integrating biological processes, so over multiple size scales. And I think you've all had, for example, courses that focus very tightly on one particular level, right? So in chemistry, you get very deep into the formation and reaction of molecules, constituent atoms. In genetics class, you focus quite a lot on the expression of genetic material from DNA to influence cell phenotypes in genetics. And what I think is really unique and really cool about this course is that you are going to develop the ability to look at a problem all up and down these various size scales. And along with that, so we'll see a first example of that today. Along with that, I also like to think about, in my own research and in many different biological systems, I think it's also really important to think about different time scales. And so on Thursday, we're going to focus much more on that aspect when we get to talking about homeostatic mechanisms. So that's coming in lecture 3. But for today, we'll be talking about integrating from molecular up to the whole body level. So one of my favorite quotes related to physiology is from the Nobel laureate in 1937, so Hungarian physiologist. I'll probably murder his name, Albert Szent-Györgyi. He was the discoverer of vitamin C. He discovered the citric acid cycle. He figured out that muscle cells have actin. So all of this, pretty recent discoveries. And one of the really cool quotes, I think, is this, describing his career. He says, in my quest for the secret of life, I started my research in histology. Unsatisfied by the information that cell morphology could give me about life, I turned to physiology. Finding physiology too complex, I took up pharmacology. And still finding the situation too complicated, I turned to bacteriology. And he really did study all these different systems. He was really a Renaissance man. But bacteria were even too complex, so I descended to the molecular level, studying chemistry, physical chemistry. After 20 years of work, I was led to conclude that to understand life, we have to descend to the electronic level, to the world of wave mechanics. But then, he quite rightly points out, the electrons are just electrons, have no life at all. And evidently, on the way, I lost life and had run out between those fingers. So, kind of a cool quote. As engineers, we'll get started by talking about today how different components build structures. So, of course, I think this is old news to all of you, but we're going to walk from the atomic level, where we're dealing with things like pushing electrons, exchange of electrons, to the molecular level, chemical reactions, chemical bonds, to the cellular level, where we get a whole host of really interesting new phenotypes, molecular recognition, electrodynamic forces become important, mechanical forces become important, up to the tissue and organ system level. So a quick review of the major building blocks that we're talking about. Well, at the elemental level, of course, we're talking about some of the most prevalent biomolecules in human or all living systems. And I think everyone probably learned that acronym, CHOPSIN, which doesn't really spell anything. If you were in high school and you learned that. Most abundant element in the body by mass, oxygen, nitrogen, carbon, phosphorus, sulfur, and hydrogen are other major components. Then you get down to down to all these important ions, chloride ion, sodium ion, potassium ion, and other trace elements, which are listed here. And how do we put those together? Well, I know you've all had a solid background in chemistry, and you know how to classify different types of chemical bonds. So here, I think it's interesting to start to focus on what are the size scales already at this level that we're talking about. So covalent bonds, here we're talking about atoms that share electrons, right? And the size scale is angstroms, a few angstroms, right, at the most. Ionic bonds, we might be talking about salt bridges in a protein molecule. Now we're covering anywhere from a few angstroms up to maybe a nanometer. Hydrogen bonds, distance is really limited to about 2 angstroms, mainly from electrostatic interactions. So down at the angstrom level. Important forces, most forces that we're going to talk about in physiology this semester are very short range forces. So whereas in previous coursework, physics and chemistry, we've certainly covered things like electrostatic forces. And you probably know in air those can be quite far-reaching. Physiology, all of that is streamed, right? Streamed by ions. So now the field that we're going to be investigating is really diminished exponentially due to ion concentration. Van der Waals forces will come up over and over again in this course. Attractive forces, again, distance of a few angstroms can be up to 1 nanometer. And hydroscopic forces, of course, in quotes now, those can act over a distance of, say, a few nanometers, chemical nanometers. And of course, those atoms assemble into biomolecules. And so if this is new, you should go back and review Chapter 2, Chapter 3, so you can become sort of, again, familiar with, able to recognize these different types of structures without having to think about it too much. So for example, here are some of the primary biomolecules we're going to talk about this semester. So fatty acids are going to come up multiple times. So you should be able to recognize features of those molecules and know, for example, that these are lipids. They're non-polar, right? So they have poor solubility in water. They come in saturated and unsaturated forms. Which one here is the saturated one? It says the top one, right? So at room temperature, which one's probably liquid? The bottom one, yeah, right, exactly. So we've got the saturated and unsaturated. Sorry, I went the wrong way. So lipids, carbohydrates. There's our saturated and unsaturated. Get bigger. Carbohydrates, the most abundant type of bimolecule, and shown here at the bottom, I showed you some 5-carbon sugars and 6-carbon sugars, because over and over in this class we're going to return to examples from glucose metabolism in the body as a case study for a whole variety of normal and pathophysiological situations. So we're going to hear a lot about glucose metabolism starting in the next 2 weeks and then continuing over the semester. That's one of the running problems you'll see again in the book, diabetes. As well, I included here the nucleotides, nucleic acids, extremely important biomolecule of energy transfer. So some examples are ATP, ATP, cyclic AMP, signaling molecules, and energy transfer molecules. We'll be hearing quite a lot about over the semester. In addition, nucleotides, nucleic acids, famously involved in information transfer as well as storage by DNA and RNA. And I'll just remind you that those nucleotides consist of a 5-carbon sugar, 1 or more phosphate groups, and a carbon-nitrogen ring structure known as nitrogen base. And these assemble such that the sugar of one nucleotide links to the phosphate of the next, creating a chain, alternating sugar phosphate groups along the backbone, identical, that chain identical for all nucleic acid molecules, while the uniqueness of the molecule arises from the sequence of those nitrogen bases. And those bases each pair to a complementary base, and of course that occurs through what kind of forces? Does anybody remember? Hydrogen bonds, right? So everybody's really spent the whole Labor Day weekend reading physiology textbook. It looks fantastic, if not a little bit unfortunate. Okay, so moving right along, nucleic acids direct the synthesis of protein, right? And so I know you didn't read this weekend because that material doesn't show up until chapter 4, but I think this is really a good time to remind you that the nucleic acids are indeed quite special in that they direct the synthesis of all the protein components of cells. And just to remind you that they do this in the following way. So the genetic sequence of nucleic acids in the DNA, which of course is coiled and buried and packaged in chromatin molecules inside the cell nucleus. It is then copied to an RNA version. That process is known as transcription, which by the way itself, that whole process involves many protein cofactors. And then this messenger RNA is processed in the nucleus and then exported. So think again of all the protein components involved. There are channel proteins, there are binding proteins, there are enzymes that are attaching and cutting various sequences. And then that messenger RNA, mRNA, attaches to ribosomes. Ribosomes are made of what? Does anybody remember? RNA and protein, right? It's a huge complex of RNA and protein. And that begins the process of protein synthesis, which is called translation. And in that process, transfer RNAs are recognizing codons of 3 nucleotides, delivering the appropriate amino acid, catalyzing formation of peptide bonds through a dehydration synthesis reaction. And then after multiple iterations, that fully synthesized protein is then able to go form covalent, non-covalent bonds. And that process, that level of regulation is called post-translational modification. So you can see we're rapidly working up the size scale here to the place where interesting cell behaviors are going to happen. So protein structures of increasing complexity, such as secondary structures like folded beta sheets or alpha helices in protein. Tertiary structures where the protein chains fold over onto themselves, bind to other intramolecular sites, perform 3D conformations that go out, do the enzymatic reaction, carry out whatever particular catalytic or structural activity is important for in the cell. Or they can form multi-protein or multi-protein complexes. That's quaternary structures. You've got multiple polymers of protein interacting together. Famous example, hemoglobin consists of 4 subunits together. And other types of post-translational modification. There's cleavage events. Some proteins are made as propeptides and they need to be cleaved to be made active. Some need to bind to a metal or another cofactor in order to become active. So there's many stages at which one might want to intervene and affect the activity of those proteins, perhaps for some therapeutic purpose. So just pause and sort of remind you where are we in the hierarchy? Okay, we just covered all of chemistry and most of molecular biology. So there you go. And now we're moving into cell biology. That was fast, right? So those multi-protein complexes comprise many of the general and specialized intercellular components in a cell. And of course, there's a vast diversity of cell types in the human body. Now, how many cell types do you think there are? I hear 100. Who thinks it's probably in the hundreds? Let's say order of magnitude, hundreds. Okay, we vote for it. Does anybody want to guess in the thousands? Order of magnitude to the thousands. Somebody, that's Connor in the middle, right? Connor, I don't know Connor. He's going for the thousands. Anybody want to go higher? No? Okay. And it depends on, maybe someone over here wants to go higher. It really depends on what's a cell type. That's a tricky question, right? If I take any population of cells and if I'm able at the individual cell level, let's imagine I have technology to measure every protein in a single cell, and I have a clonal population of cells in a dish. So I grew that population of cells into one cell, put it in a dish, expanded, now I have hundreds or thousands, and I'm going to do, let's imagine I have the technology to now measure the concentration of every protein in an individual cell. They all came from a clonal derivative. Will they be the same? No, actually, they'll be quite heterogeneous. And will they have the same behaviors? No, probably I can find some outlier cells that behave very differently. So cell types are really a cloud rather than one point in space. But if we take by gross metrics of morphology and function, there's probably a few hundred cell types in the body. So that's probably around the order of magnitude that we're talking about. But those outlier cells can be very critical. So we all know about in a tumor, there's a big debate about there's a subpopulation of cells that acts like cancer stem cells. Are they stem cells? Well, we could debate that at a different time, but they definitely have unique properties that are not like all the other cells, and they may be only 1%, but they're quite different. So they're more migratory, more resistant to treatments, more resistant to drugs, more resistant to radiation. They express different surface protein markers. They just look totally different, and very critical for the disease treatment. So, at any rate, probably around a few hundred normal cell types in the body. Okay, of course, all those different cell types in the body, hundreds of them, same genome, right? So we're talking now about the diversity coming purely from a different set of expressed collections of protein, which is really interesting. We can get back to that another day. Okay, so finally, we're at the level of cell biology. And so here is the sort of canonical view into a cell, and here we're talking about lipid biomolecules, protein biomolecules, some carbohydrates building those intracellular structures, of which you see many here. And I have to tell you that as a graduate student, the field I entered was precisely sort of a reactionary response to this grossly inaccurate image. Because in fact, when I went to grad school, it was the first time I ever heard that that cells have a structure all their own. And you see these pictures throughout your, probably since middle school education, certainly throughout secondary, high school, and probably till now. And it gives you the impression that there's sort of this, the nucleus where there's a whole lot happening with the DNA, and then there's this kind of water-filled aqueous environment, and there's some things floating around in it. And when I was in grad school, it was the first time my PhD advisor actually, I joined his lab because he was the first person who ever talked about how there was actually much more organization happening here, and that the cell was a physical structure subject to the laws of physics, and we could think of it as a physical object. And so if we look at real cells, so this cytoplasm here is not like a watery soup, right? Here's an image of some real cells, fluorescently labeled for some of the proteins that make up the cell cytoskeleton. And here you just see three types of molecules, right, labeled in actual cells. So these are labeled using an antibody or using dyes. Here the green is the microtubule proteins. The blue is staining the nuclear material, and the red is staining some cell attachment proteins. And of course, here you only see a snapshot of 3, so we're not seeing everything. There's a lot of deep dark blackness there, which you still might envision as kind of empty space. And around the same time when I started studying the cytoskeleton as a brand new grad student, I was also I went on a trip from Boston back to New York, where I grew up, and grown up outside New York City, and I was browsing in the gift shop of one of my favorite places on the planet. That's the American Museum of Natural History in New York City. And I was thinking a lot about how does information get around this cell, because my PhD project was to think about, well, when cells start to become migratory and they're going to crawl, well, they need some more cytoskeletal proteins out of the front end. And how do they get it there? Because I had been on the train to New York, and I was thinking, well, it would be like, let's see, it would be like me sending the newly made protein from the nucleus to the outer edge. It would be like me getting on the train in Boston. I need to get to some address in Iowa. That was my back of the envelope calculation. I need to get somewhere in Iowa, but I don't have a map or a road. How am I going to address the protein to this outer location in the equivalent of Iowa? And in fact, when I was in the museum gift shop that day, I found this most amazing book, which was filled with artwork created by a scientist who actually specialized in electron microscopy, and he was drawing the cell cytoplasm as a composite view to scale, all the components to scale, reflecting all the different electron micrographs that he had seen captured over the years. And so for him, the cytoplasm was not this watery soup, or this the cell was not a water balloon filled with molasses, but it really had enormous intracellular structure. It was very crowded, so lots of macromolecular crowding, which completely changes the enzyme kinetics, as you can imagine. And so I just show you some of the elements of this particular view, and he has whole books full of them. And so the largest kind of tubular structure that you see here is a microtubule. The thinner ones going diagonally here are actin fibers, actin filaments, and this knobby kind of structure going across is an intermediate filament, of which there are many, many different types. You can see it's also composed of multiple fibers have bound together. And all of this purple is ribosomes, and then, to be honest, I don't remember what the green is, but there are some nucleic acids being transported. You can see them attaching, some of them attaching to the ribosomes. So extremely dense, highly structured environment. And if you're interested in more very pretty pictures, you can go find his website or find his books. His name is David Goodsell. An appropriate name for what he does. So, cytoskeletal structures direct many behaviors of cells, not just cell migration. I was studying cell migration, but these components, the microfilaments, which are made of actin, the intermediate filaments, which are sort of neglected, but many different types involved in a variety of processes, and the microtubules, which you saw there, those are tubulin protein. are associated with control of really nearly every cell process that you can think of. So we typically associate them with things like crawling of cells, migration of cells, myosin and actin muscle contraction, movement of ciliated structures in cells, but they're really critical for all transport within cells as well as for attaching to the extracellular environment and for giving cells their structure and shape. And we can talk more about those components, but we'll have to save that for another day just because we won't get to the tissue level today. But it turns out that if you manipulate the structure of some of these cytoskeletal components, like say you pull on an actin filament or you push on a microtubule, it can really influence cell behavior. And so that's really interesting, and that's what I mean when I say that cells are physical objects and are subject to some level of control as well as chemical control and control by biomolecules. Okay, so now let's get to composing tissues from component cells. And so I think here, for our purposes today, we're going to focus on these elements of tissues. So of course, tissues, some simple tissues just consist of one cell type. Most tissues have multiple cell types within them, right? They also contain extracellular matrix. And cells are held together to each other, so we have to build tissues by putting cells together. That happens with the aid of cell junction proteins, which we'll get to in a little bit here. We also have to attach cells, in many cases, to their matrix, their extracellular environment, so not floating free, but they're interacting with what's called the extracellular matrix. And those interactions are mediated by other classes of adhesion proteins, we'll get to. And I would say for a long time, and this extracellular matrix, first of all, is itself synthesized, secreted by cells. So the cells build their own support structure, which is a really interesting engineering problem, I think. For a long time, extracellular matrix was really thought to be something kind of inert. So the histologists would look at this material on a slide, and then that region, we call that kind of the ground substance. It's not very interesting. But now we know that, in fact, extracellular matrix is actively signaling to cells within tissues. It's something that's dynamic, so it gets turned over. The different protein components change over time. And it plays important roles in disease. So, for example, my lab studies cancer. And you probably know most people don't die of the primary tumor, right? What they die of is metastases. And in cancer, it's the breakdown of the extracellular matrix by specialized enzymes, which are known as MMPs, matrix metalloproteinases. To give you a clue how they act. That breakdown is what allows cancerous cells to escape the environment of the primary tumor and metastasize to a distal site. So the changes in the extracellular matrix surrounding the tumor are a critical step in disease progression, and there's a lot of interest in trying to design inhibitors to those matrix metalloproteinases to block metastasis or to use them as biomarkers, right? So you could hopefully easily, rapidly or early on detect metastasis and injury. So we're going to be talking in this course about 4 primary tissue types in the body. Who knows what they are? You can raise your hand and call out. Now, 4 primary types. That gets a little bit too detailed, too fast. Somebody says muscular. Okay, muscular. That's one. In the back. Where? Connective tissue. Yes. Nervous. That's one. Nervous. And the last one. Epithelial. Yeah. Okay. So I know. If I was talking to my grandma, she knows what muscle is and neurons and Connective, I could probably say, what's epithelial? How do I explain epithelial? I know this is all mumble. Someone raise their hand with a really coherent and wonderful definition. How can I explain epithelial cells to my grandmother? You should always make sure you can explain your work to your grandmother.
Membrane linings?
Membrane linings. Anybody want to expand on that? Skin is one. I heard the word skin up there. Skin is an example of an epithelium. Okay, so I would say it's the tissue that lines surfaces of the body. So that's the outer surfaces as well as the inner surfaces, hollow organs. Muscle, I think, and neural is a bit easier to grab hold of. Both excitable tissue types. We're actually not going to talk too much about those today because you have a lot of emphasis on those in later sections of the course. Connective tissue, we will get into a bit more today. So connective tissue, the structural support, physical barrier, and there's, of course, multiple types. So, for example, epithelial is such a big category, right? I think we can all agree. There's just multiple different types of epithelial. So if you haven't already delved into this section of your textbook over Labor Day weekend, you'll want to go back and take a look at these because there's some great images as well as some interesting features. So a simple type of epithelium. So to give it away, there's going to be 5 types of epithelium. Okay. There's going to be 5 types we're going to talk about today. Does anybody remember what this kind is called? What category does this fall into? It's kind of a simple one. If you have an answer, you can raise your hand and then I'll know it. No takers. Okay, so simple squamous epithelium, that's not the word I want you to remember though. This is an exchange epithelium. Okay, so let's look at some of the features. These epithelial cells, thin, flat cells, right? These cells allow movement between them and through them. And in this particular case, these are cells lining, let's make sure we don't lose the power. These are cells lining a blood vessel, a capillary vessel. Okay, so has anyone grown epithelial cells in cell culture? Oh, of course you have, yes, in my lab. Has anyone grown other cell types in cell culture? In lab class or in research? What kind of cells have you grown?
Ovarian cells.
Ovarian cells, okay. And is there another?
Cardiovascular tissue.
Cardiovascular tissue, great. And have you grown cells in lab class yet? Or is that happening this year? Most of you will do that this year. Okay. And I think you will grow fibroblasts in that class. If you get there and it's different, don't blame me. But fibroblasts now look quite different from this. So epithelial cells, you know they are growing well because they are forming these cell-cell contacts, right? They like to grow in what we call cobblestone appearance. So you see something that looks like a cobblestone, immediately in your mind you say, it's an epithelial cell. And in fact, when our cells don't grow well in cell culture, they change because we either forgot to feed them or something else terrible happened. They will lose this cobblestone-like appearance and they will transition to become something more like a fibroblast cell, where they grow in a long, you'll see some later in the lecture, in a long, spindly way, disconnected from each other. And that transition is called epithelial to mesenchymal transition. You don't need to remember that now, but it's quite an interesting phenomenon in biology, and it's important in disease, in particular in cancer. All right, so simple epithelium right here. Another example, a type of epithelium, one of the 5 major types is, we heard coming back, skin. This is skin. So this is a protective epithelium. Skin is one of the protective epithelium in the body. What do you notice about this one? In fact, I have a better image. Here's a better view of the skin. So you see that there are multiple layers, first of all. Here you can see a fortified to an outer layer of matrix, which is keratin, which has been laid down by, essentially by dead skin cells. And below it, there are surface keratinocytes, which are still alive and actively producing keratin fibers, full of keratin, which is one of the types of intermediate filaments that we saw so beautifully on the good cell image of the cytoskeleton. And within this epithelium, there are cell contacts that anchor the epithelial cells to each other, which we'll get back to in a minute. There are quite a lot of desmosomes in the skin. And below that, we can even find some specialized cells like melanocytes that produce the pigment of skin. So depending on the stance, these might come out different. In your book, I think you can see an image where the melanocytes come out more brown. In this particular image, just a different stain is used. But multiple layers and this surface of deposited protein fibers characterize that protective epithelium. Another role of the epithelium we didn't get to, I didn't hear anyone mention actually. We talked about protection, we talked about lining surfaces, but a very specialized and important function of epithelium is highlighted here. Any takers on what this might be? Does anyone want to guess what this is? Just not sure, but go ahead.
Gas exchange?
This one is not gas exchange. Close. Say again?
Body.
Body temperature. Not so much in this image. So adipose tissue actually is not a type of epithelial tissue, but we will see some adipose tissue later in this lecture. So another type of epithelium is the secretory epithelium. So some types, everybody's, oh, of course, you can hear that. Oh, you can hear that, right? The secretory epithelium. There's so many types of important secretory reactions that happen in specialized tissues. And what has to happen in order to secrete protein and other proteinaceous components, right? Well, the cells have to have a polarity. They have to have an apical side and a basal side, so whatever they are producing and secreting goes to the right place, has to come out on the right side, right? So these epithelial cells are highly polarized. If we were to stain them with some, in a more detailed way, we zoom in and we have specialized markers to mark the apical side and the basal side, we could really easily detect those. We would be able to see that the Golgi are oriented, because remember, Golgi are really involved in secretion, right? They're producing the proteins, modifying the proteins that get exported from the cell. So the Golgi would actually be lined up from one cell to its neighbor, would all be pointing the same direction. So those secretory proteins are set up to go in one direction. And again, also, we often see multiple layers. And in this particular case, so this is mammary glands, this is breast tissue, these are ducts, and these are milk inside. So the white is milk proteins. Other examples of secretory epithelium are the goblet cells in the intestine producing mucin and secreting it. And if we had that image, you would also see some similar, no, not exactly the same, but similar polarized multicellular epithelial layers. Highly specialized functions. Of course, if I found some less idealized images of tissues, right, we would see multiple types of tissue in one. So here, I just left this in because here's a little hint of some stromal component in the sea of secretory epithelial tissue. So here you see milk, you see epithelium, and here you see some stromal cells, which are probably more fibroblast cells in there. Okay, moving right along. I think we took 4 or around 5 types of epithelium. So another very specialized epithelial structure is the transport epithelium. And does anyone want to guess what this image might be? Have you seen something like this before? What do you think? What organ is this? lung. No, it's not lung. Intestine, yeah, it's intestine, right. So these are columnar epithelial cells. So let's pause for a minute. If we were sitting in an anatomy class or in a medical school histology class, we would categorize all these epithelial structures by their shape. And this would be a columnar one. We would see even like semi-columnar, cuboidal, and we would just describe it by the morphology. And in this class, we're describing it, you can see, rather by the function. So here we're talking about a transport, a highly specialized tissue for transport. And what makes it so specialized? Well, again, there's an apical surface and a basal surface. You can see they're really different, right? So very specialized proteins at one side and at the other. And here, actually, did I? Yes, I did. So actually, we're going to talk more about junctions, but these neighboring epithelial cells are linked by tight junctions, and that's because components will not move from one cell next door to their neighbor cell, right? They will only channel through the epithelial layer. So it's very specialized for transport through the cells, out through the cell membrane, and not across cells to the next door neighbor cells. So transport epithelium. And the last one, I think we already heard it once, but here is an example of the last type of epithelium that's covered in your book, which is ciliated epithelium. You can see very specialized cilia structures at the edge, right? And someone mentioned earlier that the lung is a type of specialized epithelium. And that's what we're seeing here. So these beating cilia will create some fluid currents, right? Really fascinating phenomena that sweep across the epithelial cell on the apical surface, and of course very tightly linked to normal function of those cells. So you inhibit those cilia, you will inhale cigarette smoke or some other components that are toxic to those cilia. They don't work, they don't function right, and their cells no longer are able to actively move those fluids across the surface. Okay, so let's just summarize a little bit about what we said, some of the key properties of epithelial cells. So they're tightly packed. They form these continuous sheets because they cover these surfaces, outer and inner surfaces of organs. So no intracellular spaces. They're just cell layers, continuous cell layers. Usually, or actually in all cases, separated from underlying tissue by an extracellular matrix, some fibrous basement membrane. We didn't talk a lot about this, but they are innervated, although they're avascular. And they have abundant cell junctions, so those are very critical to the proper functioning of those cell types. As well, epithelial cells are often highly proliferative, and that leads to the counterpoint that they are the source of many solid tumors, right? Because when something goes wrong with regulation of that cell proliferative process, that is when a new tumor arises. And there's a question.
That third bullet innervated by avascular, does that mean yes, nerves, but no blood source?
Exactly, yeah. Yes, nerves, but they rely on diffusion from distal capillaries to receive oxygen. Okay, so since cell junctions are so critical to the proper functioning of epithelial tissues, I thought this would be a good time to touch on some of those types of cell contacts. And these are categorized in your book in many different ways. So if you haven't seen the Silverthorne text likes to really make these maps, so mind maps of how to think about material and how to use different materials. So you're going to see many different ways to categorize cell junctions in your textbook. So some of the main types that we will be talking about in this class are gap junctions, which are sort of, I think, what you think about when you think of typical cell-cell communication, direct cell-cell communication to a neighbor cell. And I'm going to show you examples of all these. Tight junctions, which are the opposite. Actually, those are occluding junctions. So they are blocking the flow of material between cells. Remember, they were important in the intestinal epithelium. And then there are what are more anchoring junctions, is what they're categorized are in your textbook. And some of those that form more structural anchoring components are adherence junctions, desmosomes, and hemidesmosomes. And I'll show you, we'll see each of them in a little more detail in just a minute. So the tight junctions first, okay, so these are transmembrane proteins fused on the outer plasma membrane. So here's a real micrograph and a cartoon, and we'll see this cartoon again on the next slide. And these typically include proteins in two families. So there's the Claudin family and the occludin family of proteins. So if you see something that's in the Claudin family, you know tight junctions are important in this tissue. And what do these proteins do? So we're just going to zoom in on that a bit more. So they fuse. So here's two adjacent cell membranes, cell 1 and cell 2. And these proteins are really fusing those cell membranes together. So this is a very tight seal between cells that prevents direct transport between them. So it's a fusion barrier for membrane proteins. And it can be found in a variety of tissues, including the intestine. It's important for maintaining the blood-brain barrier as well. So you may hear about these again when you get to the neural components, neurobiology component of this class. So tight junctions, blocking flow. Now we get gap junctions, which connect, pretty much exactly the opposite, connect the cytoplasm of two cells, and are made up of proteins that are called connexins. Connexins tend to form structures in groups of 6. So sometimes these are called connexons, because biologists are not terribly creative when it comes to naming. So groups of 6 connexin subunits assemble into this larger protein structure called connexon, and these are what form this junction structure that allows now what goes through. So small molecules can pass through, things like amino acids, ATP, cyclic AMP, a variety of ions can pass through. They can be controlled now by regulatory proteins, so at a higher level, this flow can be regulated. In the heart, they're important for synchronizing heart muscle. So when you hear gap junctions, often people think of excitable tissue because they're so critical in muscle tissue. They're also important in, for example, small smooth muscle cells, coordinating movement through the intestine, peristaltic movement. So you've got to get one cell and its neighbor to all, and they basically do the wave together, right? And how the cells see what their neighbor is doing, they would talk via the gap junctions. Another example is in the liver, so not all hepatocytes are innervated, so they might not otherwise all respond similarly to signals from the sympathetic nervous system to release glycogen, except that they're able to communicate between their neighbors, so they all do receive those signals. Found in lots of other types of organs, lung, pancreas, thyroid, ovary. That's not an exhaustive list. The anchoring junctions. Okay, so now let's talk about adherence junctions. This is a protein layer on the inside of the plasma membrane. Effectively, what it does is adherence junctions link the cytoskeleton of two adjacent cells. So they're linking the microfilaments. So rather than an open kind of channel through which the small molecules can communicate, the cytoplasmic molecules can communicate, this is the two neighboring cytoskeleton structures able to sense each other and to link to each other. So now you can imagine how this is important for structural integrity. If you pull on this cell, this distal cell also effectively feels that pull. Desmosomes, these attach to the keratin proteins, or other intermediate filament proteins, but largely to the keratin proteins, and anchor cells in that way. Related, so anchor cells, sorry, I went too fast, anchor cells to their neighbor cells in that manner. And related to the desmosomes, in fact, look very similar if you just saw them on a tissue section, are the hemidesmosomes, but instead of linking to other cells, here we're talking about a cell linking to the matrix, the underlying basement membrane of the epithelium. So again, these are made up of specialized protein structures, large macromolecular protein structures. The hemidesmosomes usually contain integrins, and you'll see that type of protein is discussed in your book. There's a whole family of integrin proteins that are mediating interactions from a cell cytoskeleton across the membrane to attach. And that's important because cells are kind of, if you want to use an engineering analogy, they're kind of spot welded onto their underlying structure, right? And so they're feeling and sensing forces and changes in that extracellular matrix, as well as actively remodeling that extracellular matrix. So it's kind of chicken and egg thing, right? And here is a sort of a close-up of some integrin molecules. So facilitating binding cells to the extracellular matrix. There is a lot of interest in designing inhibitors of integrins as therapeutic agents. That's definitely an active area of investigation. So this is one of those maps that I mentioned. I encourage you to, take a look at them, maybe make your own maps, try to understand how the functions of these different cell junction complexes are one way that we can group them. We can also group them by where they're found. We can also group them by what their components are and whether they are dealing with small molecules or if they're linking cytoskeleton and what kind of matrix they might interact with. So there's a whole host of ways to think about this families of cell functions. And I'll just encourage you to spend a little time taking a look at those. Okay, if you remember a few minutes ago, we were back talking about the primary tissue types. We were back here and we talked about epithelial cells, epithelial tissues, and we deviated a little bit to get into this idea of cell junctions being very important. So I want to come back and get to the types of connective tissue. So connective tissue is quite different from epithelial tissue, and hopefully by the time you leave here today, you'll be able to recognize them, with a split-second glance. You'll be able to tell the difference between them and whether you're looking at an epithelial or connective structure. So connective tissue is largely composed of matrix. There's lots of that matrix can be. It differs depending on what type of tissue we're talking about. So what type of connective tissue we're talking about, and we will talk about 7 types today. So it can be proteoglycans, it can be a whole variety of different protein fibers. But the defining characteristic, the first thing you'll notice about connective tissue is that most of what you see in the tissue is that matrix rather than the space being filled by cells. Right? And among that matrix protein, the most abundant one that we'll see in the body is collagen. There are many types of collagen. There are 12 different types of collagen. Another highly abundant protein fiber in the connective tissue is elastin. That's probably the next most abundant. And elastin also often interacts with fibrillin to form a different type of fiber structure, and we'll see those in just a minute. So a little more about collagen, the major structural protein. So it forms a triple helix of fibers. And then those triple helices bundle into fibrils and bundle again and bundle again. So by the end of the day, these fibers are not very flexible, but they are extremely strong. So their tensile strength has been compared to steel. They're not very elastic, but they're extremely strong. And at the other end of the spectrum, we have elastin. Sorry, cut it off. bundling into fibers, elastin really forms a kind of a web-like structure. So the